All right. So today I'm going to be talking about integrating ChatGPT into a data collection pipeline. This is something I did over the summer for NimBios, and I am super excited to present to everybody what I did. So what's actually going on here? The primary idea behind all this is that there's a ton of quantitative information stored in natural language in like scientific papers or scientific contexts. For example, it might just you might be talking about like particular events or just general data in your natural language. And if you wanted to build a data set by hand using large amounts of qualitative or quantitative information, it is tedious and it takes a very long time. So the idea is maybe we can use natural language processing, in particular ChatGPT, to automate this data collection process. And maybe it'll be faster, more cost effective. And yeah, we'll see how it goes. In this presentation, we'll be using Spruce Budworm data collection as an example. Spruce budworms are a particular type of worm and moth that they're primarily in like the eastern, northeastern US area, and as well as Canada specifically. Um, and they have this really interesting phenomenon about them where every 30 or 40 years, the population will spike. And this causes a big ruckus in the economy for like lumber or things like that, because they eat all of the leaves on the trees. And it's a big deal. And so we're going to try to collect as much data about past spruce budworm outbreaks as possible from a large amount of text. So again, here's our goal. We're using ChatGPT to read scientific papers like this from Boulanger et al. And we're going to turn it into a tabular data set like this. And let's see. So we will be using ChatGPT. We chose that over other large language models because at the time it was probably the best one. I know there are some other competitors out there, like Google Bard just came out, but at the time it was a really good one and it is very available. It's not particularly expensive to use their API and it can be set up pretty quickly. And so this collection pipeline here kind of describes how I analyze each scientific paper. So you take, for each paper, you take the extracted text, so you just get the raw text from it, and then you put it into a ChatGPT prompt, and you say, hey, okay, so this might be about it might be about spruce budworm outbreaks, but it might have information that's not relevant. So let's go ahead and summarize it with a focus on spruce budworm outbreaks. This is called a focus summarization prompt, and it's a very well-known use case for ChatGPT. After you get the output for that, which is typically a smaller volume, more outbreak information dense text, you put that into a data formatting prompt with ChatGPT. And you're saying, okay, ChatGPT, here is our summarized data or summarized information about spruce budworm outbreaks. Can we turn this into comma separated data? With the goal, of course, being to try to turn it into what is essentially an Excel spreadsheet. And then what that does is it turns it from natural language to something that looks more like data that you might see if you opened up a file and looked at all the little lines of there might be a line that says in this location in Quebec, there was an outbreak that spanned from 1920 to 1930 or something like that. And so it's just moving it into a more easily readable format for computers. After that, you put it through a data cleaning prompt, which is essentially saying, okay, we have our formatted data, but a lot of the time ChatGPT will screw it up. And sometimes two features like the location or the year might be mismatched. And a lot of the times the data that's represented in them can still be good and can still be usable. But we can't go through every single line that our parser generates when we're working with thousands of papers. So we use ChatGPT again to say, okay, here's our output from the data formatting prompt. Now let's just make sure it's all in the right format. Just switch things around if you think they need to be ordered differently. And we're just going to make it look more presentable for programmatic analysis. And then finally, you just programmatically analyze it, which is super cool. And fortunately, this pipeline worked, and I'm really happy with that. see. So there are some reliability limitations here. One thing it does that ChatGPT does, it will frequently output text in the wrong format, which is why we use something like a data cleaning prompt to try to reduce data loss where it occurs. It'll also create fake data when it's given irrelevant information, which is why this graphic down here is the way it is. There's a lot of research right now going into spruce budworm antifreeze proteins. And if you give it a paper about my parser, if you give my parser a paper about spruce budworm antifreeze proteins, it has a higher than average chance of just totally creating bogus data, which sucks, right? So how do we mitigate these types of things? There are a few ways. One, of course, is the data cleaning prompt I mentioned. Another thing you could do is to just be really careful about your prompts and make sure they're hyper-specific on what exactly the ChatGPT model is doing to your text, and just make sure that it makes sense, just like on a common sense level. Like the idea is if you can't understand your own prompt, if it's just really messy or it's telling it to do a lot of things at once and you don't really understand it, then ChatGPT will never understand it. And then finally, you can determine the relevance of the paper up front before you ever analyze it, which is super helpful and helps reduce bad data being introduced into your data set. So what we did here is we just used ChatGPT again to read all of the papers and then group them by relevance. So we're saying this group of papers they are relevant and this group of papers is not relevant. And then we just focus on the relevant ones. This is a really cool use case of ChatGPT. It's based on a type of prompt that's, it's like let's see, it's just classifying between two different options. So you're just saying either it's relevant or it's not, and that's applicable to a ton of other different types of GPT prompts when you're building a parser pipeline. And then one thing that's super important is how do we actually know if it's accurate? Like this is all well and good, but if it makes up data or it creates just bad data in any way, then that isn't a good sign. That sucks. So what we did is two of our researchers, I'm very thankful, they went through 12 papers and they hand labeled all of the outbreaks mentioned in them. And that was super tedious and I'm super grateful for their work. And then after that, we ran my parser on the exact same papers. And then we grouped each outbreak that was found. And then we compared them to say, does my parser and do these researchers agree on whether or not there was an outbreak or there wasn't an outbreak, or we think there might be an outbreak here? And so these columns represent my AI parser's predictions, and these rows represent the researchers' predictions. And you'll see that they agreed pretty well on unconfirmed outbreaks and confirmed outbreaks. So they were both able to identify the same outbreaks in the same way if they were confirmed or not confirmed in the text. But you'll see here that my AI actually mispredicted a lot of instances where there were not outbreaks in a particular location and time. And you'll also see that my AI parser never actually predicted there not being an outbreak in a particular location. And I think the reason for that is it's particularly rare for papers to explicitly mention that there wasn't an outbreak. And so it's kind of hard for my paper to use deductive reasoning to try to figure out where these no outbreaks might occur among a bunch of confirmed outbreaks. So it's super cool stuff. And then finally, I was able to run this run this parser on about 1,000 papers. After we determined the relevance, I think originally there were like 1,900 papers, but 900 of them weren't relevant. And then here's a visualization. It's just across space and time. So this represents the current year that we're looking at. And then you'll see dots representing where these outbreaks are occurring. The red ones mean that there was no outbreak, and there's only a few of those. The blue dots represent that there is an outbreak, and the yellow ones represent us thinking there might be an outbreak, but we're not sure. And then here we go. And yeah, that's the data that we collected from about 1,000 papers this summer. Then we also, I want to work on in the future, and I'm planning on it with Nina right now, creating a Python library and an R API, which will let you make a general purpose version of this pipeline so that you don't have to just make it for spruce budworms, because I don't know how many researchers are out there trying to make a data collection pipeline for spruce budworms. But I think a general purpose one would be super useful for researchers and scientists. So that's how I'd like to contribute. And then, of course, improvements in large language models like ChatGPT or like Google Bard or things like that will probably improve outbreak recognition in the future. And I'm super, super hopeful for that. And yeah, that's it.